given sufficient time, and I say sufficient time, I'm not measuring in terms of years, but in terms of months, you can remove most of the detectable glyphosate with exposure to sufficient concentrations of this consortium of PaleoRestore. In our study, we predicted that over a 6-month period, most of the measurable glyphosate would not be present. It will be degraded by the microbial consortium.
Hello, everybody. Welcome to another episode of Beyond Labels. I'm Dr. Sina McCullough, and I'm here today with Joel Salatin, and we have a very special guest. We're joined by Dr. Raul Cano, and some of you may have heard his name before. He was dubbed the Real World Jurassic Park Scientist. Today, he's going to be talking to us about how he has been working to create a probiotic using ancient microbes in order to regenerate the soil, and which would ultimately regenerate the human microbiome. Now, one thing that I think is really cool about Dr. Cano is that he was figuring out how to restore the microbiome before the term microbiome was even coined, right? So he was so far ahead of the game. I think one of the reasons why I think the work he does is so critical is because now we know that human disease is largely associated with loss of microbiome, loss of the numbers of microbes, and also loss of diversity in the gastrointestinal tract, for instance. So what he's doing, the work he's doing to basically restore the microbiome is actually going to help us be healthier, not only for the soil, like in the environment, but also as humans. He's also going to talk today about how he has discovered a way to degrade glyphosate all the way down into CO2, water, and phosphate. So he's actually able to, for the first time ever, the first person to completely degrade glyphosate. Now, as you can imagine, Dr. Cano has a very long resume and list of accomplishments. They're all included in the description of this talk. But just real quickly, I wanted you to know that he is the president and CEO of PaleoBiotica, which is a company that focuses on agricultural and environmental solutions. He has a Ph.D. in microbiology from the University of Montana, and he holds 2 master's degrees, one in fungal genetics and the other in clinical microbiology. He has earned certifications in public health and environmental microbiology as well. Okay, I'm back. Real world happened here. I had to rescue my daughter from the dogs barking and woke her up from her nap. So we're back with Dr. Cano and Joel Salatin. So as I was saying, Dr. Cano, together with Robin Steele, created a company that focuses on isolating and characterizing these ancient microorganisms from amber, deep sea cores, and other ancient environments. He's actually resuscitated dormant microbes and created a library of over 2,000 different types of bacteria and fungi. He basically applies this microbiome restoration solutions to, like we said, to restore the soil and to identify likely drilling sites for petroleum and natural gas exploration. His bio goes on and on. It's amazing. You should check it out. But I would love to just go ahead and have you hear from him. Welcome to the show, Dr. Cano.
Thank you very much, Sina. Thank you. An honor to be in the show. Thank you.
Oh. So Joel, do you want to start out with the first question?
Well, after that distinguished resume, my head's just buzzing with things. I've been very friendly with seed savers. At our home, we have a tomato. We don't know what the name of it was, but it was my wife's great grandmother's, and it's been passed down now, I think 6 generations, this tomato seed. And we still grow them every year. We keep the seeds, we save the seeds. So Dr. Cano, my first question is, how do these microbes survive? You talk about drilling and finding them in ancient places. How do you breathe life back into them?
Well, most, microorganisms in general have a remarkable repertoire of survival strategies. Some of them become so water-free that essentially it inhibits any kind of metabolic processes, most of which take place in the presence of water. So there's really no hydrolysis. Some of them have some what they call themselves resuscitation genes, which allow them to spend thousands of years in dormancy, and then once the proper environmental conditions are set or are available, the resuscitation genes will kick in and restore viability. There are a lot of organisms, even in clinical microbiology, which are called viable but not culturable. That means that they're alive, but they cannot be cultured using our techniques that we know of. So, and many organisms can be found in environments, for example, amber, that are highly dehydrating. So they essentially suck off all the water from the cell, and then basically they are in suspended animation. They are alive. All of the life processes are there, but they're not working because there's no water.
That is so fascinating, because if I'm familiar with, for example, artificial insemination on the farm, where you can keep semen in liquid oxygen, I think it is, for a long, long time. I'm also quite familiar that there are seeds, clover, for example, red clover seeds will last for 1,000 years. Dormant longevity is in lots of places. What you're telling me, though, is microbes can do dormant longevity almost beyond the imagination. Is that what you're saying?
Essentially, you also, once you look at the genetic material of a bacterium versus the genetic material of a seed, the seeds are orders of magnitude much more complex. So it becomes more of a challenge for that seed to maintain the genetic material in a functional form, as compared to a bacterium who may have 1 or 2 million base pairs.
So the simpler the life form, the longer it can stay dormant and yet be viable. So can you just give us a couple of the little, protocols in layman's terms somehow, of how you do CPR on a 10,000-year-old dormant microbe at the bottom of a gas drilling well?
Okay, well, I can give you what I do, or what I used to do. First of all, the first thing that needs to be done is to ensure that the sample you're collecting is actually free of contamination. Because it's really, if you have an organism that is dormant and a sample is contaminated with a viable organism from nature, from current or actual life, then of course the contaminant will grow faster. So first you have to ensure almost to a great extent that you're not contaminating the sample. Second, I go on the assumption that if you have been starving to death in the Sahara Desert and you fed fettuccine alfredo, the first meal, you're going to be pretty sick. So what we do is develop a protocol in which we use culture media that has all of the necessary elements, and we dilute them so that we feed them incrementally more concentrated nutrients in a culture medium that will be suitable for their growth.
Yeah,
and once that is done,
you just feed them some chicken broth.
Exactly, and with a little bit of salt, not too much fat, and basically that's it.
Yeah. Okay. All right. That's what's next.
Well, and then we, once we have ensured that this organism is growing, then what we do is try to ascertain that it's a pure culture using standard microbiological methods. And that's, once you have pure cultures, you have individual colonies that are basically clones of a single organism. Then we, unfortunately for them, we put them either in liquid nitrogen or we freeze them at minus 80 degrees. So they go from being dormant to enjoying life for a few minutes or a few days and then back into the freezer until we have the time to take them out and start looking at them. And this is what we have been doing for the last 3 or 4 years. You know, we isolated most of these organisms in the late 90s, and they were isolated, verified for purity, and then frozen because we were in the process of generating large numbers of different organisms. And then now we are going back to the what we call the culture collection and taking them one at a time and restoring them and analyzing them, both the physiological characteristics that they have as well as the genetic properties. And then we have like a database of organisms and their functions. And that's what we do.
And you've actually created a product already, right? I think one is PaleoGrow, and you've tested this on.
Yes.
Can you talk about that a little bit? What did you test and what results did you see?
Yeah, what we did was looking at this large collection of bacteria, we looked at the genomes and identified 5 or 6 bacteria that would work synergistically for a specific process. One such process would be the production of growth promoting substances so that we can accelerate seed germination as well as plant growth and the ability of the plant to take into the cells nutrients from the soil. And that we call PaleoGrow. And we also developed another similar product, but the bacteria that we selected were primarily analyzed for the ability to break down organophosphates, specifically glyphosate. And they are both similar but not identical consortia of bacteria that were genetically evaluated so that they can not only break down the product but be sustainable as a community in the soil.
Okay, let's start with the product that you created to break down glyphosate, right? Because our audience in particular knows a lot about glyphosate at this point, right? And we know how ubiquitous it is. And we spend a lot of time trying to avoid glyphosate and also get it out of our bodies. And so I find it fascinating that you have found a way to degrade it, not just to AMPA, right? The breakdown product, that you have found a way to break it all the way down to CO2, water, and phosphate. So what are your, and I know you did some initial testing. What were the results of that testing? Like how much of the glyphosate is actually broken down to CO2, water, and phosphate?
Well, if given sufficient time, and I'm saying sufficient time, I'm not measuring in terms of years, but in terms of months, you can remove most of the detectable glyphosate with exposure to sufficient concentrations of this consortium of Paleo Restore. And so in our study, we predicted that over the 6-month period, most of the measurable glyphosate would not be present. It will be degraded by the microbial consortium.
And that's what you actually saw, was most of it the majority of it was degraded over the 6 months.
Yes. And we started, with a relatively high concentration. We started with about 5,000 parts per billion. So, and that's usually, probably 3 logs of magnitude greater than you find in soil.
Wow. Well, that's remarkable. So I know if I'm listening to the show right now, my first thought is, where can I get that? Right? I want some of that product. Is it available for commercial purchase?
Yeah, actually, we are currently doing a large scale production, and I'm talking about 10,000 liters production of Paleo Restore. And we are packaging them either for individual use in terms of 8-ounce containers, or for industrial applications in terms of 5-gallon jerry cans. So the product is commercially available. We have not stopped. We have established the product process for developing, but we have not yet put it on a website, which is going to appear within the next 3 or 4 weeks. So it's not available commercially in any, and we would like to find someone who's willing to distribute our product. But right now, it's basically in our warehouses in jerry cans.
Wow. This got me to thinking, and Joel, I'd love you to chime in. So when I found out about Paleo Restore, I thought, wouldn't this be a fantastic product for, let's say, somebody who was trying to convert land that was used to grow crops conventionally, that they're trying to convert that land into like organic or regenerative farmland. I mean, don't you think, Joel? Wouldn't that be a great asset to have?
Yeah, I would think so. I mean, I would ask Dr. Cano, what do you foresee? I mean, you're really in this space. What do you foresee as the normal client or the normal customer? Is it a gardener who's just bought a little homestead and really wants to make sure their garden is clean, or is it a more commercial, farmer wanting to clean up a soybean field?
Yeah, well, the answer is all of the above. We have a study underway in California in which we are looking at basically a home farmer who has 1 acre of land that's been contaminated with glyphosate, and we provided the farmer with a free sample so that he can actually treat the soil, and of course, with a caveat that he would need to share the results. Mm-hmm. We also are working with an organic farmer, I want to say in Wisconsin, a farmer in Wisconsin who would like to get his land ready for organic regenerative farming. Mm-hmm. And so we provided him also with a free sample of Paleo Restore in order for him to test it. And we're, based on what we've looked at in our laboratories and field studies, I'm very hopeful that it will be a successful endeavor. And therefore, between our better manufacturing processes, as well as the more supportive data that we're generating, that in fact, this would be primarily a tool to restore soils for organic farming, or even restore soils for better yields.
Sure. So I know that a lot of soil can be, or toxicity in soil can be ameliorated simply by bringing up the organic matter, getting the humus up, and with composting and getting a lot of organic matter up. I call organic matter the Alka-Seltzer of the soil. It's a buffering agent. Speak to the whatever the difference between, for example, buffering it out with heavy applications of compost versus using using Paleo Restore?
Well, the composting process, actually what it does is increases the organic matter. And I presume that in parallel with this infusion of organic material, you're infusing it with water. And therefore, what you're increasing is the microbial diversity of the soil. And where the serendipitous or by intention, they would be microorganisms in the soil that would, and there are microorganisms in the soil that will break down glyphosate. This is more of a trial and error situation. In the case of Paleo Restore, it is a combination of microorganisms that, in fact, have been shown to break down glyphosate. So it's a more sure thing. But when we apply it in an aqueous form, so we're introducing water as well. And since the microorganisms are grown in the presence of molasses, we are providing the microorganisms sort of like a nutrient kickstart so that they can become established in the soil and then participating not only in restoration, but also in plant feeding. So that's basically as I see it, the difference between the two processes or the two methods.
So that's fair because I know that that high organic matter, if you can build a lot more organic matter in your soil, it just does a lot of ameliorating work in a lot of things. And this is the clever speak of some of these chemical companies. They say, well, it degrades in the soil, but it only degrades in soil with high organic matter, and so this is kind of the wordsmithing and the clever speak. That the fact is most chemical fertilized fields are very low in organic matter, or certainly active organic matter. Not necessarily legacy organic matter, but currently organic matter, and so they don't break down very well. Sina, I know you're dying to ask a question. Go ahead, Sina.
Yeah. Okay. So listening to you two talk back and forth was really intriguing, because I'm thinking, so do microbes, modern microbes that we'll find in the soil here, like say at Joel's farm, at a regenerative farm, are there microbes now that can break down glyphosate? And if so, then why spend like the money to go back to get the ancient microbes to have them break down the glyphosate? Are they more efficient at it, or what's the difference between using our current microbes that we have at our disposal versus the ancient microbes?
That's a loaded question. I
finally got my daughter off my lap, so now I'm in the conversation again. I'm thinking like.
Yeah. Well, yeah. Go get your daughter back. No, really, the situation is the following. You're looking at two different set of organisms that have been exposed to modern environmental stresses and organisms that have never seen it. So you're looking at two different genetic pools. And presumably these organisms are able to survive in a broader space, broader spectrum of environmental conditions than they are now. They're being selected for just a specific environmental set of stresses. So even though both groups of organisms are capable of breaking down glyphosate, that's not the issue. The issue is have them break down glyphosate and still survive and thrive in the soil environment. And what I'm saying is that probably there's a greater genetic diversity in the ancient microorganisms than there is in the modern counterparts.
Okay, so if that's the case, then they could be, for instance, more likely to survive different stressors that they might experience. So in that sense, they could be more efficient because they might have a higher survival rate once they're
Correct.
in the soil. Okay.
And there is another aspect of it. Microorganisms in nature function as guilds. There's a group of coordinated effort to achieve one specific function. It could be, maybe achieved via cross-feeding. So the administration to the soil of a single organism that has a specific function, for example, break down glyphosate, that organism has a relatively low probability of survival when it's actually going to be introduced into a new ecological niche and had to compete with those organisms that are established. So I think even though they might be able to break glyphosate in the laboratory, it is not certain that they would be able to, as individuals, function in the soil. When we prepare PaleoGrow, we not just have one single organism. We have a microbial guild of organisms that are cross-feeding, and therefore they do not have to compete with other organisms, as an individual, but they have actually an ecological niche of their own that once introduced into the soil are less likely to be outcompeted by the organisms that are already present in the soil.
Okay, but so now you're talking about a different product, right? Now we're talking about PaleoGrow.
No, PaleoRestore is the same thing.
Same
PaleoRestore
concept.
is a microbial guild. Yeah, the entire company product line is based on the fact that microorganisms in nature function as guilds. And therefore, we prepare products in the form of guilds that are created based on their genetic composition rather than on their name. Because, if we look at a probiotic bacterium, Lactobacillus acidophilus, we all think that it is a probiotic organism. In fact, there are many strains of Lactobacillus acidophilus that have absolutely no probiotic value whatsoever. And some of them are extremely good. So it's just not just the species, but the strain. And that's really very important to emphasize, that we actually select our organisms for the guild based on their strain composition.
Yeah, that's a good distinction. I'm glad you brought that up. And I also want to just bring up that even though, people call you the real life Jurassic Park scientist, and you're talking about isolating these strains, you are not genetically engineering anything, correct?
Absolutely. In fact, our whole mantra is natural. We even grow our organisms in nutrients that are organic, certified organic nutrients, because what we are trying to address is a natural process by natural organisms that have not been intervened in any way by genetic engineering or even genetic manipulation. And I will go one step further and say that the way that when we isolated these organisms, the first isolation we call generation 0. And then when we actually have it purified, we call it generation 1. That generation 1 was propagated, created generation 2, and those were the ones that were frozen. So every time we go to make a new product, we go to generation 2. So it's as close to the original strain as it could possibly be. So little or no genetic changes have taken place from the original strain. And that's really a process that, is time consuming, add cost to the process, but it ensures that all the products are genetically homogeneous.
So in the soil, so the other product, PaleoGrow, I've got a question about why these microbes went into hibernation. I mean, if they were soil based at one time, you would think on the whole planet that something would have survived in the soil somewhere if they have such survival characteristics. I have a follow-up when you deal with that, but deal with that and I'll have a follow-up.
Well, this organism, there is a continuum, okay, of organisms that were in nature, let's say 2 million years ago, and they have been multiplying and generating new generations, developing new generations of bacteria over that period of time. But some clone or some group of them got trapped, for example, in amber, and all of a sudden that particular genetic generation got frozen in time. It just became a Kodak moment. And the same thing with some of the soil organisms that get washed into sediments into the bottom of the ocean. They stopped reproducing, but they didn't lose their viability. So they had stopped evolving by either a physical or a chemical process that took place many years ago. And some of their counterparts did not stop, and they continue growing on and now we call them modern organisms. But all of these modern organisms have somewhere along the line an ancestor.
So my follow-up then is that those microorganisms that you're finding in, for example, natural gas wells, those, when they were viable up on the planet's surface, they accounted for much of the ancient flora and fauna. I mean, we know, from mastodons to dinosaurs to whatever, and there had to have been a lot of lushness that we can't imagine today. Is that right?
That is basically the composition of the ancient environment in which they encountered. Well, actually, it is ancient to us now, but to them was contemporary.
Sure, yeah.
So they represented the microbial diversity, at least a slice of the microbial diversity that existed at that time, where the dinosaurs or the mastodon or whatever geological era we are looking at. So yes, but then if they stopped genetically evolving because of a physical or chemical phenomenon, then they sort of became a representative slice of that time. And then those who were not, entrapped in any way, they continue to evolve, and they comprise now. And they're evolving along with the composition of the soil and the environment in which they live. So you can have a continuum of genetic diversity or genetic undiversity over time, but you have these bits and pieces of entrapped Kodak moments in the evolution of life that are entrapped in some sort of, like, for example, amber.
Well, it's like the mastodons, as when Siberia thaws and they're finding these mastodons with grass in their mouths. Obviously, yes. that wasn't tundra at one time.
Yeah, exactly.
Well, we're about out of time.
No, go ahead.
Fascinating. We're about out of time. Sina, do you have another burning issue here before we close out?
I do. So I have two follow-up questions because I can just see people in the audience wanting to know this. You talked about the Paleo Restore to break down glyphosate in the soil, and the Paleo Grow, right, to help build micronutrients, to help build microbial diversity in the soil. Is there a product for humans, like a human probiotic that you're working on? And Can the Paleo Restore be used in water to break down glyphosate that it would be in the water?
That's let me answer the second question first. I think it will require some sort of bioreactor in which the Paleo Restore is in some sort of column, so the water passes through it, and the microorganism will be working on it. In fact, we filed a patent about 2 years ago, which is in process, describing a process in which you could treat water contaminated with glyphosate using some sort of a bioreactor that we have described. So the answer to this, yes, right now, just in concept. So that will require a good engineer to help us out to work this out. But I think that's definitely a possibility. And it would actually be, I suspect, even more effective because there's already water. You don't have to water the line. And the other question, a probiotic for humans. The answer is yes. We have, not us specifically, because we specialize and focus only on environmental microbiology, but the company, the BioCollective, has a product. I believe they call it Sugar Shift. And in the Sugar Shift, there is at least one organism that I know of that has the capacity to break down glyphosate through an alternative pathway. So in that respect, there is a product that could work. I don't know that he has ever been tested. The PaleoBiotica, very unlikely we do that because most of our organisms are not what we call generally recognized as safe by the FDA. So you cannot consume it. So in other words, it's called grass. We don't have grass organisms. We have organisms that grow on grass, but it's not grass organism. But the BioCollective not only has this organism, but has the wherewithal to create one such product.
Okay, and my last question, because I know we're trying to wrap up here, is you've already come amazingly far in your work, and I think this is remarkable. Like when we first talked several weeks ago, I was just bouncing around the house, excited because I have not heard anybody doing this up to this point, especially with the complete breakdown of glyphosate. So how can we help you to make this more successful? At this point, are you good to go? You have everything you need? You can get this out to consumers, or where are you?
Well, there are several things. First of all, money talks, and of course, this last production batch, it basically is a bet on a roulette wheel that this is going to be useful and everybody's going to want it. So that's an investment that we already made. But I think the most important thing, as far as I'm concerned, is to have a market, individuals who are interested in purchasing the product for us and help us distribute it. So in other words, the marketing and sales aspect of the company. Right now, we are focusing on the production, and I think we are already past proof of concept. So we took a chance and manufactured 10,000 liters, which is a lot of acres. And we have now a contract with the manufacturer to produce it as we want to and do some product development. But the idea is to sell the product. And if we have a partner who can help us do that, even better.
So you need a partner to help you?
Yeah, basically with skills in marketing and sales and distribution. That's basically.
Okay, so
That's
if
every,
anybody,
that's every inventor's nemesis, isn't it? You
It
have
is.
they have these genius inventors, but then finally you need a Zig Ziglar, a Zig Ziglar to go put it in the marketplace.
Exactly. There is a lot of competition, and the only thing that we have going for us is that we are all natural, and probably the price point is going to be lower than current chemical fertilizers right now by a large margin. So, but we don't have the wherewithal for large scale distribution and sale. That's basically it.
Right.
so if somebody is listening and they're interested in partnering with you, how could they get a hold of you? Do you want us to put your contact info in the description box for them to reach you, or how would you like that to be handled?
That would be perfect. The contact information in your my personal information. Yeah, that would be wonderful.
Okay.
We have numerous multimillionaire listeners.
Not me, one of them.
Well, Sina, let's wrap up. This has just been fascinating, and I'll let you wrap it up for us.
Yeah, I think this is just remarkable what you do. This could really propel us forward, right, in this movement of trying to have a clean ecosystem, clean food, healthier bodies. I mean, this is a solution that I'm actually very excited about. And I think a lot of people listening are excited about this. If anybody's listening and wants to partner with Dr. Cano, I mean, he's a good bet, right? I mean, we're in the presence of greatness here. And Joel, I know a lot of scientists, and you probably never heard me say that before, but yes, he truly is not an inspiration and just brilliant. So we'll put the contact information in the description box. You can get a hold of him. And then Dr. Cano, when this is available for everyday people like me to purchase, will you let me know and come back on the show and tell us how we can get it?
Yes, I will. In fact, we have there is a website, and I'll share that with you, BiotiQuest website, that we have something called Yield and Shield. It's a product that is a combination of PaleoGrow and PaleoRestore that we prepare specifically for the BioCollective so that they can sell it through the website as Yield and Shield. And it comes in 8-ounce containers.
Fantastic.
And I will send, yeah.
Okay, great. So we'll post that too in the description box. So if anyone's interested, they can try it out. And if you do, let me and Joel know what you think about it, right? Email me at handsoffmyfood@1791.com. Dr. Cano, this has been so inspiring and enlightening. Thank you so much for coming, and we can't wait to have you back on the show again.